The $1,000 Genome was a successor of sorts to my previous book called Cracking the Genome, which covered the race for the Human Genome Project. And I've been struggling to really think of an idea for a popular science book that would build on the Genome Project, in part because I couldn't think of a hero or an anti-hero to kind of frame a story. And all my ideas sounded too much like a textbook, which I didn't really want to write. And then in spring of 2007, I watched a webcast of Jim Watson, the guy who co-discovered the double helix in 1953, receiving his full genome sequence on a portable hard drive. He'd been selected as a guinea pig by 454, which was the company that first launched a commercial next-generation sequencing machine. And I thought that was a pivotal moment, that someone could actually receive their full genetic code neatly embedded on a small computational storage device and actually learn something from it. I thought that was important, and likely Jim would be the first of many, many, many people to be sequenced. And just a few months later, in fact probably just a few weeks later, through a mutual friend, Chris Anderson, who at the time was the editor of Wired magazine and had been a colleague of mine at Nature, I was introduced to Linda Avey, the co-founder of 23andMe, and she told me about their plans that were publicly announced later in the year to launch one of the first consumer genetics companies, direct-to-consumer genetics companies, where individuals for a relatively small amount of money, a few $100s, could get access to their own personal genome information in terms of genealogical information and risks for genetic diseases, genes they may carry for various disorders. And again, I thought that was democratizing genetics and was a really exciting development. And so the $1,000 Genome really was a sort of a mix of those two trends and how advances in genome technology would impact the public, both who learn they have genetic diseases and perhaps preemptively want to learn something about their genetic makeup. 2013 has been the 10th anniversary of the Human Genome Project. I was flattered to be invited to join a symposium at NIH to mark the 10th anniversary, which was great fun. The landscape is really unrecognizable from where we were just 10 years ago, in large part, because we've now got such phenomenal technologies around us. We have these machines from companies like Illumina and Life Technologies that are essentially genome centers in a box. The power, the horsepower of a machine that now sits in a lab, one of these machines can mimic, if not exceed, the throughput of a genome center 10 years ago. And so any lab now is virtually its own genome center. And the power of that is playing out in academic research with the ENCODE project, for example. We're now able to tease and assign function to huge tracks in the genome. We can do microbial sequences at a pace never before imagined, finding the cause in rapid, almost record time for foodborne illnesses. And in the medical arena, we're seeing huge advances. We're seeing the ability to now sequence children with rare, undiagnosed disorders and end their diagnostic odysseys by sequencing their full genome and finding telltale mutations in a number of cases. And in cancer as well, there are now companies like Foundation Medicine getting off the ground. In fact, that company just went public recently, that can screen 200 or 300 important cancer-related genes in a tumor sample that could be referred to them from anywhere across the country or around the world, and you can pinpoint critical mutational information in that patient's individual tumor that can in turn be used to figure out which drugs might best tackle that pathway that's been disrupted in that patient's tumor, as well as clinical trials that they might enroll in. So wherever you look, the new technologies, new sequencing technologies are playing a dramatic role, and I think we're on the verge of seeing that also play out in the pharmaceutical industry as we really turn over a new leaf in drug discovery as well. The ethics of genome sequencing is, how long do we have? a full hour. I think just in the past year, less than a year, we've seen many groups weigh in on this. There was a big study in Science magazine at the beginning of the year from Yaniv Ehrlich and colleagues at the Whitehead Institute, showing it would be possible to look at anonymous samples in a genomic database and actually pinpoint or triage the identity of some of the contributors to a database by looking at markers on the Y chromosome, pinpointing a surname, because the Y chromosome is inherited through the paternal line, just as surnames are in most countries. And so Yaniv's paper, which was published in Science magazine, really woke people up to the possible ability of people to unmask anonymity, and that is something that clearly needs to be looked at.